Good evening, everybody, if you're in the UK, and very happy and delighted to speak to you today about the work we've been doing on RNA vaccines, initially for COVID, but also thinking of this technology for future pandemics and other health indications. And what I hope to give you in this talk this evening is really a sense of where we came from, what the last 18 months to 2 years were like, and where we're heading to. I hope that whatever your background, you'll be able to get something out of this talk and understand a bit more about how RNA vaccines work and how they're really changing the way we think about vaccines and global access to vaccines in the future. So I used to start with this slide back in 2018, before COVID had arrived. And I first used this slide at a presentation at the World Economic Forum, saying, if there was an outbreak of a new pandemic and you were standing in a major city somewhere in the world, how quickly would you be able to access a vaccine? How quickly would you imagine that a vaccine could be generated? And it's interesting, in 2018, there were about 30 people who turned up to the presentation. And if you ever go to the World Economic Forum, you can see who logs into your presentation. And they were all the spouse of, the partner of. So nobody was really taking it very seriously that there might be a future pandemic. But in fact, pandemics are not new. And if we think about the historic number of pandemics that have gone on in the world, really they've been around since man started to live in high density populations. Going right back to the Justinian plague, which was the Black Death, although not named that at the time, which killed over half the world's population and may have been more instrumental in bringing down the Roman Empire than the invading hordes. And so very interesting how plagues have shaped our history. And then, of course, the Black Death killed a quarter of the world's population. If you ever go to Siena, which is a beautiful city, go and visit the cathedral there, and you'll see that it's only half complete. And the reason it's half complete is they were halfway through building it, and the Black Death killed 2/3 of the population of Siena. At the time, they didn't understand about pathogens, and they thought they'd been too ambitious and God was punishing them. So they left the cathedral unfinished as a testament to ensuring that they weren't over ambitious in the future. And you can see, so it goes on, many different plagues, particularly in South America, that were brought to the New World by the European invaders. Moving to the Spanish flu, killing 50 million people, and HIV/AIDS more recently, which I have spent most of my scientific career working on, killing over 37 million people, and still with about 600,000 deaths per year, year on year. So nothing new, but we were not particularly well prepared. And we live in a world where there is increasing threats of more epidemics coming through, partly because we are encroaching more on natural habitats, and many new emerging viruses can cross from animal species to humans. But also we're starting to see a higher threat of antibiotic resistant organisms arising, and those will increasingly become a potential threat to mankind. And in that context, it may also be possible to develop vaccines that may have some activity against the antimicrobial threat. And then the final thing would be potential bioterrorism, which still remains a possibility. And the access to genetic manipulation means that essentially this type of work becomes easier as time goes by. Well, let's think about vaccine success And it's always nice to go back to the roots and think of smallpox, that is the one virus that's been eradicated through vaccination. And of course, we're very aware in COVID-19 of vaccine hesitancy and anti-vax movements. And of course, that's also nothing new. This beautiful cartoon was taken around the time of Jenner's life, saying that if you got vaccinated against smallpox, you'd probably turn into a cow, because of course the vaccinia virus from the cow that was used to prevent smallpox was obviously related to cows, and that's why we have the word vaccination, vaccinia or vacca, and that's why it came out that way. Now, interestingly, some people have gone back and looked at the original vaccinia strains, and it looks as though they were probably isolated from a horse. So maybe we should be talking about equination rather than vaccination. But smallpox killed over 500 million people in the 20th century. And you can see this rather disturbing picture now of two children, one who had smallpox and one who got vaccinated and was protected against smallpox. And that's the kind of evidence that anti-vaxxers really don't like to see. Vaccines clearly work. And so far, in terms of a medical intervention, it is actually the most successful medical intervention, the most successful public health policy beyond access to clean water, and has so far prevented over 3 billion disease cases and greater than 500 million deaths. And you can see in that nice graphic on the right-hand side how it's been incredibly successful against a great number of diseases. And this causes a problem, because the more successful vaccines are, the less memory people have of the diseases that they created. And so the fear of side effects from vaccines is far greater than the fear of the disease that the vaccine is against. It's interesting to recognize that in many parts of the world where some vaccines are not available, particularly measles, mumps, and some of the pneumococcal vaccines, there is very little hesitancy about vaccines if you can't get hold of a vaccine and you still see diseases ongoing. But one of the problems is that vaccines are a global good, and it means that in terms of the marketplace, most of the vaccines that are used globally are actually targeted at a price of less than $1. And so this has stifled a lot of investment in developing new vaccines, because commercially it is less attractive. Well, along came COVID-19, and everybody was wondering what would happen. And now I'm going to talk to you a little bit about our experience of working on COVID-19. So many of you will remember the start of the pandemic, and we were sitting at Imperial hearing about some strange cases of a new virus occurring in Wuhan in late December. And by the end of the year in 2019, there were 27 cases reported. 7 of those were serious, 2 were recovering, and people were really still unaware as to how serious this pandemic was going to be. Well, of course, we then saw a very rapid explosion of COVID-19 in China, but then a very rapid rapid containment of the infection within Wuhan and the surrounding community, because the Chinese government were very able to restrict the movement of people and put in self-isolation, distancing, the use of masks, and actually controlled the epidemic in China very rapidly. But of course, the genie was out of the bottle, and people had already traveled from China and seeding infection in the rest of the world. Now, just thinking about that experience in Wuhan in China reminds us of how effective some of these very basic implementations are of blocking transmission. We know in terms of social distancing how well it has worked to reduce infections, and this really models your risk of infection depending on the level or distance you are from somebody who is infected with COVID-19. And this nice graphic at the top really reminds us how effective masks are, because they both block the level of viral shedding and reduce acquisition. And it's still surprising that it's quite hard, given that there are still a serious number of infections going on in the UK, to encourage the general population to continue to wear masks in high density environments. And in fact, this other graphic is extremely useful for explaining how risk versus occupancy impacts transmission rates. So you can see in the right-hand part of this graph how low occupancy outdoors is really relatively low risk, particularly relatively low risk, even if not wearing a face covering. But as soon as you get into poor ventilated spaces, the risk goes up. And in high density spaces, the risk goes up very dramatically, particularly with no face covering down in this bottom sector. Well, what about COVID-19? Well, essentially, like many new epidemics or pandemics, it had a viral reservoir. And you'll hear much debate in the media about whether it really came from animals or whether it was a lab outbreak. But by and large, the majority of the scientific community believe that it was definitely transmitted initially from bats through possibly a secondary animal reservoir and then to humans. And this can be tracked forensically by looking at the genetic sequence of the virus as it has changed from bats to humans. So I think we'll never be able to prove certainty that it was not involving a laboratory, but I think that that's the least likely scenario. Well, we went from a few cases in 2019 to a global pandemic. And now currently, as of today, there are 245 million confirmed cases of COVID-19 and 5 million deaths. And you can see the density of cases on this nice global map. Of course, that's probably likely to be underreporting because those are confirmed cases, and there may be many more cases that have gone undiagnosed. Well, I'm now going to talk to you a little bit about RNA technology and how that's been used to develop vaccines. And so I want to step back a little bit just to remind you about how proteins are made from your DNA. So all of you are hopefully remember that DNA resides in your nucleus, and that's what your chromosomes are made up of. So that's your inherited genetic material. That sits there and is unchanged. And in order for that to be converted into a protein, a process of transcription takes place where the DNA is translated into RNA. And RNA is this intermediate nucleic acid that comes from the nucleus into the cytoplasm of the cell. And essentially it has the message that encodes a protein structure. So for every protein that a cell needs, it will have a different message. And then it uses this ribosomal machinery, which is essentially the protein making factory that reads that genetic code and makes the protein. So when we think about the flow of information, it goes from DNA in your chromosomes to RNA, this intermediate, into protein. Now, one of the advantages of RNA is it has a relatively short life. Your genes, obviously, you don't want to change, but in order to control the amount of protein you make, the RNA has an inbuilt decay mechanism so that after a while, it naturally decays and you stop making that protein. If you need to make the protein continuously, you just make more and more copies of the RNA so that this process continues. And that gives you the very tight genetic regulation. Now, what we do with RNA vaccines is essentially take this RNA component, and rather than it being encoded by the nucleus, we actually make the RNA synthetically and introduce it into a cell so that it can engage the cell's machinery to make the protein that's encoded by that sequence. Well, we had been working on this technology at a very early preclinical stage for a few years before COVID-19 came along. And the reason that we were excited about the technology is it allowed us to use what we call a synthetic production process. So rather than using animal cells, cell culture, animal products, this can be made fully synthetically in a relatively small manufacturing site and had the potential to make millions of doses within weeks. Now, at this stage, nobody had made millions of doses of an RNA vaccine, and it had not been proven to work in clinical studies. Well, once the Wuhan epidemic started to take off, one of the really important things that the Chinese researchers did was that they made the genetic sequence of the virus available online so that everybody around the world could access that genetic sequence and use it to start developing vaccines. And that occurred on January the 11th. And that was really the starting gun for people to think about whether they were going to make a vaccine. We were sitting at Imperial, and I'm based on the St. Mary's campus in Paddington. And I had a discussion with two of my postdocs saying, well, should we make a vaccine against this new virus? And one of them said, I don't think it's worth it. You know, we've seen lots of viruses come and go. It's in China, it'll probably disappear. And the other one said, well, maybe it's worth a shot. And I sat on the fence until the 19th of January. And then we thought we should try and make a vaccine. But at that stage, we thought this was a vaccine that, if we worked really fast, might be used in China, not necessarily for the world. Well, what we did, as many of the other companies working on RNA vaccines also did, was to take the virus and select from the virus the genetic code that it actually encodes the sequence for the surface glycoprotein, the spike that sits on the coronavirus. So rather than using the entire genetic code of the virus, we just isolated that sequence and used that as the basis of the vaccine, produced that in manufacturing facility. And what the next stage was to encapsulate it in a lipid droplet to prevent the RNA from degrading and to be able to deliver it as a vaccine strategy. Well, this is the code of one of the RNA vaccines that's out there. This is the Pfizer-BioNTech vaccine. And you can see the genetic code here on the right-hand side of the screen. And the black letters are the exact sequence for making that spike glycoprotein. And the blue sequences are specialized structures that go on the front end and the back end of the RNA to give it stability. And essentially, you can make this in a small test tube, or you can make it in a large manufacturing plant, and you use a DNA sequence to make lots of copies of the RNA, and that RNA is then used in the vaccine. Now, you can go from a genetic sequence to assembling it as a DNA template, and then you use that template to make your RNA and package it in these very sophisticated lipid droplets. And that, in a nutshell, is how you make a vaccine. Now, of course, there's a lot of skill and more detail in that process, but what it allows you to do is to be able to make between 0.5 to 5 million doses in a liter of reaction material. Now, that's very different to conventional vaccines, which would require thousands, if not tens or hundreds of thousands of liters to make the same number of doses. So you can see, hopefully, how this is a very attractive technology for moving fast and making vaccines at a cost-effective fashion. Well, most of the vaccines that are out there, particularly the two that have been authorized from Moderna and Pfizer, essentially use genetic material that encodes the spike glycoprotein. We call that an antigen. That gets into the cell, and then it expresses that glycoprotein. What we at Imperial have been working on was a slightly different process, where we have not only the antigen that we're interested in, that glycoprotein, we also encode what we call a replicase. And this is an enzymatic system derived from another virus that allows the RNA to be copied. So that when the RNA gets into the cytoplasm of the cell, it's amplified up and you get far higher levels of expression of the antigen. And the advantage of this means that potentially we can use much lower doses to get the same response, but also we can get a longer term level of expression. Well, this is a nice cartoon of how the vaccine works. So I've told you already that essentially we take just that genetic code for the glycoprotein, that spike on the surface of the virus. We inject it into the body and it's taken into cells where it interacts with the cellular machinery. So there's the RNA going into the cell. It then starts to copy itself, and you start to set up lots of these little microfactories churning out the protein that express the glycoprotein on the surfaces of the cells. And because that protein is foreign, it's not part of you, the immune system recognizes it as foreign and starts to make antibodies and cellular responses. So rather than manufacturing complicated proteins in a large manufacturing facility, we're actually harnessing the body's own protein machinery to generate the vaccine itself. So if you like, we're getting your cells, when you have a vaccine like this, to do the manufacturing job for us. So the way it works is you have the vaccine, it's formulated, it's injected into your muscle, it's taken up into the muscle cells and into some immune cells. It starts to be amplified, it expresses that foreign protein, and that induces the immune response. Well, what does it look like? This is a very short graphic showing the RNA in the cell, encoding those glycoproteins and inducing the antibody responses. And what you see that happens is those antibodies respond to that foreign protein. You induce those antibodies so that when you see the virus, they can interact with that same spike protein on the virus and either destroy the virus or prevent it from infecting target cells. And this seems to work relatively efficiently. Well, we moved very quickly to test our vaccine in animal models. We needed to know whether it actually worked because our technology had never been used before. And we were very happy to see that it produced very good levels of antibodies that were able to neutralize the virus. And this gave us a lot of confidence in moving forward because we thought, right, if this works well, even at extremely low doses, hopefully it would have a chance of working well in humans. So we had to pull a lot of teams together to move essentially from designing something over 2 days on a computer, getting it assembled into a genetic construct, assessing whether it worked in small animal models, and then understanding how to manufacture it. So we had not manufactured this vaccine, and unlike the large companies that have already been making vaccines, we had to find a manufacturing company that were able to generate our vaccine to what we call a good manufacturing process suitable for use in humans. And you can see we were able to move from January to June to getting it into first in human trials. So this was faster than we had done at something like this before, not as fast as some of the other companies, but we had to do a range of toxicology studies to demonstrate that our vaccine was safe, whereas others were able to jump straight into humans because they had already tried their technology for other vaccine indications previously. Well, it was a very intense period, and we were able to move this first vaccine into human studies, initially in Imperial, but then at other centers around the UK, and recruited willing volunteers to come in and take a vaccine that had never been tested in humans before. And obviously to do that, we had to work very carefully, doing what we call a dose escalation study, where we start with very low doses that we didn't think would work, but to check on the safety. And so we went cautiously, building up the number of participants as we went. So we started our first in human study in June. By October, we were then expanding into a couple of hundred participants, looking to see whether our vaccine would be effective enough to move into large-scale efficacy trials. Now, to put this in context, we had no idea whether it would work. We only had animal model data, and all the other candidates out there had already had some proof of concept against other targets. So we were working very blindly, having to move at speed and make a number of choices that we might not have done so quickly had we not been working in the COVID-19 space. A very intense period of research with a great number of researchers at Imperial literally working around the clock to make this happen. Well, the results of that early trial were slightly disappointing, in that we had a black and white situation. The majority of the participants made good antibody responses, and these antibody responses were equivalent to the level of antibodies that you see with natural infection. But we had a number of participants who failed to make any response at all. And so that was really very puzzling for us and prevented us from moving into large-scale efficacy trials. So we already had worked up trial sites around the world to go into a 20,000 person study, but we had to call a halt on that because this was not meeting our pre-prescribed requirement for moving into those large trials. So we had to go back and understand why were the animal models misleading us about human immune responsiveness. Now, what we have understood more recently is that, in fact, human responsiveness to RNA vaccines is very much dependent on your innate reactivity to the RNA itself. Now, RNA obviously is the basis of many viruses, and your immune system is hardwired to make what we call an antiviral response, an interferon response. And if that's too high, it essentially shuts down the protein manufacturing capacity in your cells, and you get no vaccine take. And so we've had to now retune our vaccine to sit in this middle place where we get better expression and less innate immune response. We've done that recently, and we're now in the middle of a second trial in the UK. We hope that that will make a significant difference, and we're rolling out a new trial in Uganda as we speak. We hope that that will start in the next few days. So we learned a lot from that very rapid and intense period about the technology, and that's really accelerated our development of something that's completely new by about 5 years. Well, why did we not do as well as the others? And essentially one of the issues was cash. So we were doing this with a relatively modest budget, funded by the government and also by philanthropic donations. But compared to the large pharma companies, you can see they were all investing billions of dollars. That meant that they could try many different versions of their vaccine, and we had to put all our eggs in one basket. Now, of course, it's great that all that private money went into those vaccines, because essentially we saw those vaccines coming through and they're now being rolled out in the global community. And hopefully many of you will have received some of those vaccines and maybe even a third booster. Yeah, you can see the relative size of our budget and what we were trying to do to compete with large pharma partners. Well, I've talked a little bit about the RNA-based approach, and ours is slightly different to the Pfizer and Moderna vaccines because our RNA self-amplifies. We still think that that has very important potential advantages for the future. But the other vaccines that are out there are those that are produced by viral vectors, and this would be equivalent to the Oxford AstraZeneca vaccine, where they use an adenovirus, which is inactive, but carries that same genetic code. So they're packaging the genetic code in a virus, and that delivers it to the immune system in the same way as our genetic code is delivered, but in lipid droplets. And then more recently, we've seen some vaccines, particularly the Novavax vaccine, that actually uses manufactured proteins to induce a good immune response. They have been slower to get to the end game because they've been more complicated to manufacture. And the vaccines that are out there look incredibly successful. So very high levels of efficacy. You can see that they are up in the 90% for some of them, down to 50%. Now, at the beginning of the pandemic, people were predicting that if we got a single vaccine candidate that was as good as the 50%, we would have done extremely well. And you can see, as a global effort, we exceeded that 50% expectation. But cost is still important. And while we see that the RNA vaccines worked very well, the RNA vaccines made by those commercial companies are of high cost compared to some of the others. And so you can see, for example, our colleagues working in Oxford with AstraZeneca, their vaccine is significantly cheaper. And so we're seeing far higher numbers of these vaccines being used in low and middle income countries where access and affordability is really important. Now, one of the things that I think is often lost in people when we think about these approaches is, what are these vaccines actually doing? And all of the vaccines and those numbers in terms of percentage effectiveness is based on their ability to prevent symptomatic infection. So when we say a vaccine is 95% effective, a COVID vaccine, it's 95% effective at inducing disease. It is not 95% effective at blocking infection. And so while we see currently there are still a high number of infections occurring in the UK, which is troubling, you can see that actually the amount of disease is significantly lower than it was this time last year. So we know that these vaccines are modifying the epidemic and are preventing serious infection and hospitalization. We still do need to see more vaccines being given to those that have been resistant to getting the vaccine in the first place. And we still don't know how long vaccine immunity will last. And that's why we are seeing the rollout of booster vaccines, trying to see if that will help prolong immunity over the winter period. And I suspect as we look into coming years, particularly those of us who are over 50 are likely to see that we will be getting annual vaccines for COVID for the foreseeable future, most likely alongside flu vaccines. So we have yet to see a vaccine that actually prevents infection with COVID-19. That may be realized sometime in the future, but I think the general public still is confused by the fact that they've been vaccinated, but they're still potentially a source of infection. Well, it's been a great success in the UK in terms of the number of vaccine doses that have been given. But when you look at it globally, it's a very different picture. So essentially, the world is now divided to those in high-income countries that have had high levels of vaccination in 2021. You can see in the lighter shade of blue, those that will get to the same coverage in 2022. But you can see a large swathes of the world are unlikely to get to decent levels of coverage until 2023. And so, if nothing else, this pandemic has again alerted the world to lack of access to essential medicines and vaccinations and an inequitable state. A lot of effort is being done to try and resolve that. But as we move out of this pandemic, I hope that people will start to look at building new capacity to change that dynamic. Well, I've told you about RNA against COVID. We're still working on our platform against a wide number of indications, some of those that are listed here. And what's really nice about this technology is essentially you can plug in any protein you want to make. So today we're working on COVID-19. Tomorrow there might be another outbreak of a different Ebola strain. We just change what's in the genetic code and we have a vaccine against a new and emerging strain of Ebola. So this is an exciting approach and technology that is changing the way we think about making vaccines. So when we look to the future, what might the future look like for future pandemics? Well, I hope I've shown you that we've been able to go from a genetic sequence to now being able to design vaccines on a computer and creating a prototype. This new technology also allows us to move away from the current paradigm where you have very large manufacturing facilities in developed parts of the world that then ship vaccines to other parts of the world. What we're already seeing now is a movement to start to produce very small manufacturing plants across the globe and in low-income countries, so that should a new pandemic occur in future years, instead of relying on a few manufacturers to ship around the world, we may be able to see that the same vaccine could start to be made simultaneously in every region of the world and increase much greater access to vaccines in the future. Well, you may have thought that we are out of the race because we were unable to compete with those large pharma partners. But what we have been able to do by racing in the COVID-19 epidemic is demonstrate that our technology has real potential for the future. So from Imperial, we were able to set up at the beginning of this year, a new company, a spin-out company from Imperial called Vaxxer Equity. And the name is important because it's looking to provide equitable access to vaccines. And then very recently, this gained a partnership with AstraZeneca, who see the potential of our vaccine technology, although not having won the race for COVID-19, is likely to be the technology of the future because it can deliver much lower doses, lower cost, less frequent dosing, and because of the low dose, can be very amenable for delivering combination vaccines. So we're already looking at combinations in the context of influenza and COVID-19 or against pan-hemorrhagic viruses like Ebola, Lassa fever, and Marburg. But also this technology, we're starting to realize, not only works for vaccines, but can be used to encode many different proteins. And so not only are we looking at infectious disease, we're starting to look at its use in cancer, in protein replacement, and many other health indications. So really, we feel that we're just at the start of this technology, and the future years really are looking very promising for realizing this type of approach for many different disease indications. So I hope that's given you a really nice view into vaccines, what they can do, the technology we continue to be developing at Imperial, but also a little look at some of the other vaccine approaches and some of the challenges moving forward in terms of providing global access to vaccines in the future. And thank you very much for listening, and I'm delighted to take any questions. Alan, you're on mute.
I'm on mute. I hope I'm now unmuted. Am I? Can you hear me?
Well, I can hear you now.
Great. Well, thank you very much, Robin. That was quite a tour de force, and a very exciting one at that. So thank you very much. That was a super talk. So we have some chat questions. One or two of them look as if they have people have had trouble with the screen. But one question from Piers Lesser: can an RNA vaccine cross the blood-brain barrier? And if so, what would be the consequence?
That's a great question. So the vaccine approach that we're using at the moment, we deliver into the muscle, and we know that actually it travels very short distances, pretty much contained within the muscle, and what we call the localized lymph node that makes the immune response. So it doesn't get into the systemic compartment, and it wouldn't cross the blood-brain barrier. There are approaches using the technology that, by others, actually, where they want to look at systemic distribution for other indications. So one might want to actually look at delivering RNA into the brain if you want to change the protein chemistry. That's something possibly for the future. But in terms of the health risk, there is no possibility of it crossing the blood-brain barrier.
Right, okay, thank you. Now we have another question from Philip Mincham. Philip, we have the possibility, if you would like to unmute yourself and switch your video on, you could ask the question yourself rather than have me repeat it. Are you visible? Here he comes. You're still muted, Philip. And also your video is pointing at some indeterminate shape. Can you unmute yourself, Philip? Well, let me go ahead and ask you a question for you, if I may. Philip's question is, could you make a new polio vaccine by this method, or for all of the usual childhood jabs? And would there be some advantage to that?
Yes, I mean, theoretically, we could make vaccines using this technology against all the childhood vaccine targets. But actually, in reality, they work so well, and they can now be produced at scale for such a low cost that it wouldn't really be sensible or worthwhile to do that. Now, the one exception probably is polio. And the reason that is an exception is that as we are getting close to eliminating polio, there has been a move away from using the live polio vaccine, because the live polio vaccine always has that potential that it could revert back to being pathogenic. And so if we're truly going to eliminate polio, we need to use non-replicating or infectious polio vaccines. So it could be a case for using an RNA vaccine against polio in the future.
Okay, thank you. Now, here's a question from Helena. Helena, are you around? If you can hear me, you might want to unmute yourself and ask your question. Where's Helena? No, I don't see or hear Helena. Someone has got his hand up, a gentleman there. I'll come back to you, sir. Well, Helena's talk is, Helena's question rather, is thank you for a brilliant and exciting talk. Is there a risk of creating double-stranded mRNA, which may be cleaved by adding the replicase in the construct? Could this reduce the vaccine efficacy to create immunity?
So that's also a nice question. And we take great care in the production process to make sure there is no double-stranded RNA. But RNA itself is relatively fragile. And that's why the current RNA vaccines are kept at very low temperature. And that is definitely a drawback. And that will be a drawback for their global distribution. But what we are doing to try and get around that is actually make essentially freeze-dried vaccines that will be temperature tolerant and can be stored at room temperature. Because one of the things that causes RNA to decay is it's hydrolyzed. And if you take out the water, then it's extremely stable.
Hmm. But I guess you would then have to allow it to reabsorb water to become effective again.
Yes, but there are a number of vaccines where you add sterile water for injection to rehydrate the vaccine before its use.
Right, I see. Okay, thank you. Right, I'm not having much luck at getting people to ask their questions so far. Richard Stock, if you're there, would you like to ask your question? Let's try one more. If you can unmute yourself and switch on your video for good measure. Let's give Richard 5 seconds. I feel like saying fingers on buzzers, everyone. No sign of Richard so far. Richard wants to ask, is there any possibility of an autoimmune reaction being triggered?
So that's always an interesting question, and autoimmunity is a very complex science. I think that the chances of triggering autoimmunity with an RNA vaccine are probably lower than other vaccine candidates, but you can never exclude it altogether. Of course, the chances of triggering autoimmunity by COVID-19 are much higher. So it's in terms of the risk, it's always relative risk. The risk of triggering an autoimmune response to natural infection is so much far in excess to any vaccine triggering an autoimmune response.
Right, thank you. Let me go to a gentleman who I think had his hand up. Sir, Mr. Tekeste Teki, did you want to ask a question, sir?
Yes. It is intriguing for me that the infection numbers are rising 40,000, 50,000 nowadays, despite double vaccination, boosters, and so on. I believe partly it's because there are people who are hesitant to take the jabs. Now, what can be done to reassure those who are hesitant to take the jab? From your lecture, it is safe, but what makes them not take it? What can be done?
There's no easy answer to that question. And, I think about some of the talks that I was giving at the beginning of the pandemic, and people were saying, how will you ever get up to 70% of the UK population vaccinated? And people being very skeptical as to whether that's possible. Well, we're now at about 85%. It may be very hard to persuade people who are not open to rational debate and who are absolutely opposed to vaccination to take it. There are also some people who still remain concerned and frightened about vaccines, and others who feel that it is not a threat to their health, particularly those who are of a younger age. So I think that will always be a challenge. But it's also important to recognize that these vaccines are less effective at blocking transmission. And so because they don't block transmission completely, the virus will continue to circulate probably forever, in the same way as we have not eliminated influenza. And that's why I suspect we will see booster vaccines being used on an annual basis for many years to come. But I don't have a magic solution for getting that last small part of the population that are resistant to the vaccine, taking the vaccine. All we can do is explain the science, explain the risk benefit, and continue to try and make a good case.
Thank you, Robin. A question from another of our members, Peter Michael. If you're there, Peter, and want to ask your short question, please do so, if you can unmute yourself.
Thank you. Yes. Riveting. You make it sound so simple. I was just wondering, what was the little iPhone-looking gadget that was in the sort of bottom left-hand corner when you were talking about the RNA construction?
So that's a reaction vessel. It's a bit bigger than an iPhone. It has a volume of about 250 milliliters. And from that, we can make several tens of thousands of doses. And essentially, it's an enzymatic reaction where we put in a DNA template. We put in the nuclear bases that are required to assemble the RNA, and an enzyme that copies the DNA into RNA. And that's why it's a synthetic process. And that can be scaled up to several liters and produce millions of doses.
Amazing. Thank you. Thank you. Now, next question from Faizan Tariq, one of our major donors. Thank you. Would you like to unmute and ask your question? You're still muted. Would you like to ask your question? No. I have
Alan, I'm using somebody else's laptop, so my name is Azad Ayub.
I thought you were in disguise tonight, Azad.
But my question is, where has it gone? Well, how do you know in practice when it is time to give a booster?
That's a good question, and it's not an exact art, because everybody will, after their first 2 doses, will have reached a different level of response. Mm-hmm. And it's a bit like rolling a ball up to the top of the hill. Once you've had your second dose, your immune system hits the highest response and then starts to decay. And each individual will decay with a slightly different rate. And so people have modeled the level of immune response and see that at about 6 months, it's dropped to a point at which it's still likely to be protected, but that you would get the maximum benefit of a boost. But that's done on an average population level, because you can't do that on an individual basis.
And does it depend on the size of the person?
No, doesn't seem to be size dependent.
Right. Okay. Thank you very much.
Thank you. And that was Azad Ayub, one of our major supporters. Thank you, Azad. I thought you were in disguise and having a non. Robin, could I ask if you might tell us a little more about the company you set up and the collaboration that you're embarking on with AstraZeneca?
Yes, well, I mean, I think one of the things that I learned really through the last 2 years was that while we spent a lot of time in the basic research, it was very difficult to develop a vaccine without major pharma support. And we looked at it early in the pandemic and everybody was too busy and our technology was too early. But having got it to a level of testing in humans, we were then able to track the funding to set up a small spin-out company. And Imperial is very good at really translating their research so that it can then be exploited. And then in discussion, in fact, AstraZeneca had been watching what we were doing and had seen me give a number of presentations, were then also very interested in the potential of this technology for the future. And that really gives us now a base from which, rather than trying to do something purely on an academic setting, it would have a global reach, should it reach its full potential. It's still at an early discovery stage. And while I'm an enthusiast, I'm also a realist that we still have quite a long way to go to make it really game-changing.
Yes, and perhaps they're seeing the potential scope for a broader range of applications than the current version of COVID-19, at least.
Yes.
Yes, okay, understood. Now, I think I missed a question that's popped up. Where is it? From Mr. or Mrs. Lai What to everyone. Are you still here? And if so, would you like to ask your question yourself by unmuting? You're down there. I'll ask you to unmute. Right. I've got another name has come up on that box. Please go ahead and ask. It's this person here. You're still muted, I'm afraid.
Okay.
That's it.
Right. In one of your slides, you showed a number of viruses which you can develop the relevant vaccine. I noticed that dengue fever wasn't included in one of your items. Is it because it's a totally different approach or what?
I mean, there's clearly a need for a very good dengue vaccine.
Yes.
And it has been a challenge. It's been a challenge because of the different strains of dengue and cross-reactivity between the different strains. I think that will be solved, not necessarily by us, but it would certainly be something that we would like to try the technology against. So it's not as easy as COVID-19 to solve, or we'd already have a dengue vaccine, but I think that we will see more research in that area.
And then chikungunya is easier, is it?
Yes, chikungunya would definitely be an easier target to go for.
Thanks very much.
Thank you for the question. I have read actually that one reason offered for the absence of major investments by big pharma into many of those more exotic diseases like dengue and Ebola and so on, is that they can't see themselves making much money out of it and can't to recoup their investment necessary. And perhaps your technology, Robin, might offer an avenue for doing that.
I think it will, or we certainly hope it will. And the other advantage is because you can make things at small scale and quickly, it does change some of those commercial equations. And so, there are some infections that are geographically, contained, and so they're not a global market, and those have not been attractive to large pharma. And it may be that the economic argument for this new technology makes those more realistic. So we think that it will change some of those approaches and make them more tractable and more commercially attractive. But there are some things that still remain difficult because there's such a big scientific challenge that the risk of investment versus return is not favorable. And examples would be, HIV, for example, and even dengue. Sanofi spent hundreds of millions of dollars on their dengue vaccine that didn't come up to the level of efficacy that would make it a very marketable product. So sometimes it's not just whether there's a market or not, it's also whether the science is sufficiently mature to say your investment in going after that is going to lead to a product that is marketable. So the two things are important.
Well, good luck with all of that and with your future research efforts and with your quasi-commercial efforts and collaborations, Robin. And I'd now like to invite our chair of Friends, Professor David Southwood, to give a vote of thanks, after which perhaps we can all unmute and give you some applause. Thank you again. David, over to you.
Thank you, Alan. And thank you, Robin. I thought it was a magnificent display of, well, it was very exciting. I wanted to know, you took us through your adventure. Clearly, there were disappointments, but on the other hand, I sort of feel a sense of satisfaction at the end. And I am sure you've transmitted that sense to me, and I hope to the audience. For me, I'm lost in admiration for the manner in which you tackled something that matters to everybody. It changes human life on this earth, potentially, with a new approach, new technology. And I think you've really put over to us what a joy it is to be a scientist. And I'd like to say for myself, thank you very much. And I would like everybody to unmute in order that we can give you a stirring round of applause. So, Robin, thank you.
Thank you very much.